Welcome to Two Bees in a Podcast, brought to you by the Honeybee Research and Extension Laboratory at the University of Florida's Institute of Food and Agricultural Sciences. It is our goal to advance the understanding of honeybees and beekeeping, grow the beekeeping community, and improve the health of honeybees everywhere. In this podcast, you'll hear research updates, beekeeping management practices discussed, and advice on beekeeping from our resident experts, beekeepers, scientists, and other program guests. Join us for today's program, and thank you for listening to Two Bees in a Podcast. Hello, everyone, and welcome to another episode of Two Bees in a Podcast. Today, we are joined by Dr. Ashley Mortensen, who is a scientist for the Plant and Food Research Group, the Institute of Bioeconomy Science in Hamilton, New Zealand. Hey, Ashley, thanks for joining us on this episode.
Kia ora, everyone. It's great to be back.
Yep, we had you in the past. And, Ashley, I'm really terrible at this. I forgot what we discussed, but I'm sure it was amazing. And we brought you on this time to discuss physiological responses of colonies to apiary population density, which when you emailed us like a couple of different options, I'm like, well, gosh, I want to talk to her about all of those things. But this is the one that rose to the top. And so we're going to spend some time chatting with you about this. But before we do that, and I know you've been on before and folks know you, it's still good to remind our listeners who you are and how you got into the honeybee world.
Yeah, that's a great place to start. And it's a story that I like to share anyways. So nice to make sure that anybody new hasn't heard it, or anyone I'm new to, I guess, that hasn't heard it. And then also just to remind everyone, because it's a bit of a non-traditional path into a research science role and into honeybees. I was actually always grew up liking animals, was told, oh, you're going to be a vet. And I didn't want to be a vet, so was a little unsure for a while. Ended up being a zookeeper. So I worked with primates and big cats and moved around with my partner at the time and ended up working at a vet clinic as a vet nurse and picking up wildlife rehab. And through wildlife rehab, met some people who were keeping bees. And I'm a serial hobbyist. And so anything new to learn and try, I was like, yep, I'm going to keep bees now, and took actually an extension course through the South Carolina extension program, and then won a beehive for scoring the second highest in the test for that course's year, and caught a swarm immediately afterwards. And then 2 years later, was moving from South Carolina to Florida and emailed you, Jamie, about how to move my beehives. And then worked in a research lab as a technician and decided I really liked research and I really liked bees and I wanted to do something more. So then I came to you for graduate study in honeybee research and haven't left yet.
Okay. Sorry. I am laughing because I'm like, you never left Jamie's side. Okay. No, let me start over again. Sorry. I'm like, you haven't left the honeybee world, not
fair. Well, left. I guess That's I haven't really left Jamie all that much either, because I'm in monthly meetings with you at like 1 a.m. New Zealand time.
That's true. Yeah, we're both on a committee, an international committee together. We do both meet at times that are inconvenient for both of us, but convenient for many of the other members on the committee.
Well, before I go into asking you a question that we were going to ask for the interview, I do want to say, Ashley, this has probably been brought up, 4 or 5 times in our podcast. And it's the story of Jamie going out to do field work with you on that
We talked about this
day.
On the episode I did, because it was about the DCA project, Jamie, and you sitting by that dead raccoon during the lightning storm.
I was like, this is me. The dead raccoon is just what I'm about to be.
Jamie probably never goes out in the field with grad students now because
you've
ruined it.
That's enough.
You're done.
It kills me every time it gets brought up. And I'm sure if we had like a Two Bees in a Podcast trivia, that would be one of the most memorable things, I think. So
it's even better that it's on like Disney World property as well.
Was that on Disney? Oh, gosh.
Yeah.
It was a long time ago.
All right. Well, I wanted our listeners to, be reminded and reintroduced to you, Ashley, in that, dead raccoon scenario with Jamie. All right. So we are interviewing you today about physiological responses of colonies to population density. So basically, the number of colonies in an apiary. Can you elaborate what this means and maybe provide some background behind the project that you're working on?
Yeah. So when I first moved to New Zealand, one of the first big questions that was posed by beekeepers, and I've also heard it in conversations in the United States, but specifically around honey production and Manuka honey production, where so many people are trying to take colonies into these distinct areas to collect as much honey as possible at the same time, was what does overstocking look like in honeybees? And in New Zealand and in the Waikato where I'm at, dairy is a huge industry. And so, we have a fairly good understanding of what type of grass or plants you have out in your paddock, how many cows you have in your paddock, what that nutritional intake looks like, and how that relates over to how much milk they're going to produce. And that stocking rate, and then when you tip over into overstocking and you start to lose productivity per animal. And so what does that look like in a honeybee hive? The more we thought about it, the more mind-boggling it becomes, because the landscape that every colony is in and what forage is available and what pollen versus nectar and the quality of that and the density of that and what other animals are in any given environment, and how could you possibly come up with like a number that is accurate to more than just this one specific location, and how do you practically get something for beekeepers that helps them to start to understand what's a good stocking rate versus a bad stocking rate in an area before the end of the honey flow when they realize they made a lot of honey or not. So one of the ways we started thinking about that was, well, what can we look at in beehives in real time? Where do we see indications from the bees themselves that they're experiencing stress due to population density? And would some of those things be something that over time we could refine to be able to, obviously it's not as good as knowing this is how many we put in there, but while you're there in the first couple weeks, is there some way to look at it and be like, oh, too many hives, and do something about it before you find out at the end of the season? So we've kicked that around a little bit and then are trying to come up with what those indicators are. And right now they're maybe not super practical for use, but you've got to understand what they are to be able then to refine to something that's like a usable tool.
Yeah, so when you posited a couple of options for us to discuss with you on this podcast, when I was looking at this particular project idea, it was intriguing to me, right? Because commercial beekeepers can stock a lot of colonies in a yard or a few colonies. And I routinely get asked a few times a year, if I'm keeping bees in this area, how many colonies can I put per acre and how far apart do they need to be? And you think about things like honey production, stuff like that, but you guys are looking specifically at physiological responses. Our listeners around the world are going to think, well, what does a physiological response mean? And then which ones did you look at and why?
Yeah. So the fun thing about a beehive, which you'll know everybody, and this is something that you've really, hooked me into from the beginning, Jamie, is the concept of the superorganism, right? So you have the individual honeybees and kind of how their bodies are functioning and performing. And then you have the colony itself and how that overall is functioning as an organism. So it's really, I think, fun in bees that you get to focus at both levels. We've done a little bit of looking at both, but the colony is where my real interest is. And so I had one master student a few years ago who looked at things of the individual bees that might indicate. So she looked at metabolic rate of the workers, and so kind of how much air they're moving and how that relates to their function. She also looked at glycogen reserve, which is like the amount of basically like energy that each worker bee had stored in their body as adults and as immature larvae and pupae. And then she also looked at heat shock protein across individual bees in apiaries at different locations. But the one that I guess I was planning most to talk about today is that colony level one. And that one, we're actually looking at drone production and how drone production changes with population density.
So Ashley, I would love to hear more about your study design and how you measured the responses, of drone production. I'm very intrigued by this.
Yeah, I think it overlays with a lot of interests of mine. I think it's a pretty charismatic idea to talk about. And so obviously the last time I was on here, I talked about drones and DCAs and collecting drones at drone congregation areas when I did my master's study with Jamie. So it really is kind of my heart place in the bee world is fun things to do with drones. I think they're pretty just silly and goofy and often overlooked in their role besides going out and mating, but what they represent and what they indicate in terms of colony function and colony maturity and stability in the beehive. And when I was still studying at the University of Florida, Jamie was obviously my lead advisor, but Dr. Christine Miller was also on my committee. And her lab does a lot of things in male-male competition, reproductive competition, in other types of insects and other bugs. And there was a seminar speaker who came and he was talking about reproductive competition between males. And so he was saying that when you have increased population densities in populations where males have to compete for mates, we know of things like having big antlers or protecting territorial patches and things like this that you recognize in maybe like deer or in a lot of different birds. But they also, a lot of different species, you'll actually see increased sperm production in the males when they perceive that there's increased reproductive competition. So if they're able to mate, they have the best chance of being successful and being the one that's actually like fathering the offspring. And so when you think about a colony of the bees, so obviously we have drones and those drones themselves produce sperm. But as the colony as a whole, the drones kind of individually represent the sperm of the colony, each one of them. So we've kind of joked before that they're like flying gametes. And so if you look at the colony level, do we see changes in the amount of drones that they're producing based on the population density? And outside of reproductive competition, you might think if there's a whole lot of competition, you're in an area that has way too many bees in it, probably nutritional resources are going to be limited to that colony. You're not going to have as much food. You'd assume that something like drones that aren't contributing to the work that's being done in the colony, they're not going out and collecting food, they're not taking care of brood, they're not building comb, they're not defending the hive, that you would reduce your investment in those because they're pretty big. They have a long larval period. Like they require quite a lot of resources to raise. And my favorite things are things that feel a little counterintuitive as well. So we were like, if we looked at colonies, would we see something where they reduce drone production when they're in a density stress situation? Or would we see this pattern of reproductive competition where if they're in a high population density, where they actually increase drone production, and what would that look like or mean?
Okay, so that's really fascinating. You've said a lot of things that I'd love to follow up on, but I'm sitting here thinking the whole time you're talking about this. So you're essentially interested in looking at, does colony density influence total number of drones produced per colony?
Yes.
Okay. And so how did you set up that study and what did you find?
So I guess the biggest limitation to say out front is obviously quite an effort to set up a spectrum of apiary densities in terms of having enough colonies. So there is low replication. So all of this that we're going to talk about from here would be considered as like a case study or preliminary study that just indicates if the hypothesis is sound and worth following up on. So
good caveat, Ashley.
We set up 3 apiaries. The first apiary only had 8 colonies in it. The second apiary had about 60 colonies in it, and the third apiary had at least 120. There was a little bit of flux over the year, so I think it went up to like 150 at one point and went back down to 120 just because we only have so many spaces that are ours to set colonies up in, and we had some colonies that came back from a trial and needed a place to hang out for a while. And those numbers, obviously that 60 apiary in the middle is still kind of a high number. But when we'd done the other work, looking at individual worker bee responses with a previous master's student, 40 was our middle apiary. And we weren't quite seeing differentiation between the 8 and the 40 apiary. So we just pushed it up to 60 so that we would be able to have 3 points that we were assessing across of to look to see if any one apiary was just an anomaly or if it seemed to be meeting a trend. And then in each of those apiaries, we had 8 colonies that were our focal colonies. And all 8 of those colonies had been established that spring. They had new queens. They started in a single 10-frame brood box out at the same location. And then they were all transported to their 3 different focal apiaries and allowed the period of 1 brood cycle before we started any assessments on those 8 specific colonies within those other apiaries. Each of them was given one of the plastic drone cell frames that was partially drawn out. I mean, ideally they would have been fully drawn out, but we've had a couple seasons now of trying to get all of our beehives to draw out all of those frame for us, and they're all maybe like 75% drawn. So we gave them a frame of drone brood, but we also gave them a top bar with no foundation, no comb on it at all for them to decide what type of comb they were going to draw on that. And then the rest was standard worker cell for the other 8 frames in that hive box. And so then we went out and we quantified the amount of drone brood on any frame in the colony with a little grid that we overlaid over each frame, and specifically noted how much drone brood was on that green drone cell frame and how much drone brood was on that drawn wax frame. We also quantified how much drone cell had been drawn on that wax frame and how much worker cell had been drawn on that wax frame as well. And we did that at 3 time periods, 1 mid-spring, 1 late spring, 1 early summer. And then an additional at the early summer point, we collected drones during the mating flights at the entrances of each of those colonies, and then also looked at sperm viability in those individual drones.
Huh. What were the results that you found from this?
Yeah, so excitingly, and also in a way that kind of ruffles a few people's feathers, we ended up seeing that the high density apiary appeared to be drawing the most drone comb and producing the most drone brood. And then the low density one was producing the least drone comb and the least amount of drone brood. And then the middle one kind of wafted around, but it still like fit the trend. So it was just really exciting. And there's obviously the caveat that it's low numbers. They're in 3 different apiaries that are in 3 different foraging landscapes, but all 3 of them were foraging landscapes that were Waikato haddock adjacent. So it's a lot of clover and grasses, maybe a little bit of blooming shrubs nearby. And all of them were areas that there weren't any known apiary sites nearby. And actually that high density one was right here by our work building. So we know the area really, really well in terms of what was nearby. And then the 8 apiary one was in the driveway next door to my house. So we're pretty confident on what the landscapes are that they were in as well. And it really appears that there is this variation in the amount of drones produced, with the ones that were in those really high density colonies seeming to produce more drones in response to presumably reproductive competition at the colony level.
So actually, that's really fascinating. So I'm sitting here thinking to myself, and you've given all the great scientific caveats to your local conditions and the number of colonies and blah, 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 right? The stuff that we would all say interpreting any of our stuff conservatively. But let's just say that this trend remains true. And if you did it again and again and again and again, you found that colonies in high density apiaries produced more drone comb and reared more drone offspring. Let's just say that trend that you've seen is true. How would they, like, what feedback are they receiving that, I mean, I know you've thought about this, right? And maybe you don't have to share if it's trade secrets and you want to
No,
say
it's not trade secrets. It's more questions.
What are you thinking? Yeah, what are you thinking here about how they would perceive that? What information they're using to do this?
Yeah, so I mean, obviously the biggest thing you think of when you're overstocking or oversaturating area is that it would be something related to nutritional intake, right? And obviously they ought to be able to perceive that there's a lot more competition at floral resources. But it seems really strange for that to be what would be feeding back to modify drone production. But one of the things that is on the docket of further things to test looking forward would be to be able to set up that smear of apiary densities, but then provide supplemental feeding so that we know that all of them are having all of their nutritional needs and feedback just fully covered in a really neutral way so that we could eliminate that variability from it. There's still, of course, a lot of logistical things to figure out and overcome as to how to do that without just creating like robbing mania and a whole bunch of other problems in your apiary, but some way to ensure that you're eliminating that variable in terms of just food consumption. And I mean, maybe it doesn't necessarily have to be eliminated, because if that's the answer, then that's the answer, but to be able to at least tease that out. One of the other things that in earlier conversations with Dr. Lee Simmons at Western Australia had looked into, or had a postdoc really cursorily kind of do a preliminary trial on, was looking at smell. So they tried to pump the smell of other colonies into bee colonies to then make them think there's more hives around or not. That was only with 3 or 4 colonies, so there's not really clear data to come out of that. But obviously those odor stimulants are reasonable. And then also we've done it at an apiary because it's easy to manipulate an apiary scale. But what's the radius away from that colony that is relevant to? So I have more questions than answers for you. But it's a really fun brain teaser to really think through. And this is the place that we're at with it right now, is actually in this enjoyable but daunting brain teaser phase.
As you're going through, I'm just thinking of all these questions. And of course, I'm thinking like with honeybee research that we just kind of sit here and think about logistics, of like how we can put together a project and like, how do you know that that's actually what we're looking at, right? There's so many factors and it will keep us all up at night, I think, thinking about that for too long.
Yeah. So the thing I didn't mention yet, that's also another like fun little add to it, is that in the individual drones, when we did the sperm viability analysis from these colonies at the different apiary densities, the sperm quality of the drones in the higher density apiary was less. They had a lower percent of viable sperm than the drones in the lower density apiary. So you do start to see where potentially they're making more drones to have those greater numbers out at a DCA to compete with mating with a queen. But in doing that, you are compromising the amount of attention, care, nutrition that can be delivered to any one of them. So you saw a reduction in the sperm quality in those drones, but there were so many of them out there. And in the world of honeybees, where you have single mating, there's so much, like the sperm volume from any one drone is as much or more than any queen actually stores in her spermatheca. So really like the number of viable sperm maybe isn't as important because she's going to mate with upwards of Tarpey's not recent anymore, it's been a while, was like upwards of 50 drones. And so it's more important to have the numbers there to successfully mate potentially than to have that really high sperm viability, which is a trade-off that you see in other species as well. So in other species, which is why we check the sperm viability, in other species where you see this increased sperm production, it's not uncommon to also see a decrease in the quality of any individual sperm as well in that kind of numbers game that's happening in this male-male host copulation reproductive competition.
So I recently just took an insemination course not too long ago, and I've kind of like delved in and I don't know, I just have more and more interest in drones these days too.
Because they're super cool.
They're very cool.
And their big eyes are hilarious. And they're so clumsy flying around. Like when you're sitting at an entrance trying to like count drone comings and goings or catching them and you're just watching them come and go from the hive, I'm like, there's nothing not to like about you guys.
Yeah, they're pretty great. That leads me, well, that doesn't lead me to my next question, but
I'm going
to ask you yes, I'm going to ask you the next question. And that is, what implications do you think your results have or might your results have for beekeeping?
Yeah, that's a tricky one in the applied space as well, because what beekeeper going out into a high production scenario is like, let me leave 2 frames of open space for drone production. So again, as I said at the beginning, it's not necessarily the most immediately applicable, but it is us starting to kind of unpeel that onion to figure out what type of responses are we able to find, and then what would that look like as we got further and further down to like a quick assessment. One of the things that Epernay also looked at was that heat shock protein, and there was also some variation in the heat shock protein levels across apiary densities. That heat shock protein did show some variation across population densities, which is not actually about heat. It's a stress response protein that's synthesized across a whole bunch of different species in response to physiological stress. And so that's one of the things that showed some promise even in the worker larvae that a previous master student looked at, and then also seemed to show some promise in this drone study that Epernay had done as well. So finding some of those things that are more achievable, because you could collect a sample and send that off rather than having to give your colony a frame of, quote, wasted space to make drones, and then just have to go back and look and be like, no, do I have more than 1 frame of drones? Let me do something about it now. Because of course, when we talk with beekeepers about this, they're like, this is the most impractical thing ever. And I'm like, yeah, I haven't said it's practical yet, I promise.
Yeah, Ashton, so that's one of the things I think, we've got a lot of beekeepers from around the world listening to us, and they're always looking for that applied angle. What can I gain from this? But sometimes it's like projects like this that start you off in the right direction that ultimately lead to that information. So I think you've talked about this being a lot of student work. You've got a lot of ideas and more questions and answers. So I'm assuming you guys have some follow-up projects planned.
I mean, I have follow-up projects planned. I just also have to get the funding for follow-up projects planned.
Of course. Spoken like a true scientist.
I mean, I have an indefinite number of projects planned, Jamie.
So Ashley, that's fascinating. You've re-spiked my interest in drones. I mean, I know they're not just those lazy guys that only have a mating purpose. I'm sure there's some great value to it. Maybe that will be one of your lasting legacies is allowing us to be more sympathetic to this bee in the hive that doesn't get a lot of positive press, right? So thank you. Thank you for continuing to work on this topic. It's really interesting. I can't wait to see where your team heads with this.
Thanks. Yeah, it's really fun. So it's exciting to get to share it with people and just have a bit of time enjoying the novelty of it instead of worrying about where to go next.
It's everybody's favorite game show, Stump the Chump.
All right, welcome back to the question and answer segment. We have something unique going on the next 3 or so Q&As. We're going to do a 3-part series on OA, oxalic acid. And the reason we're doing this, to be quite honest, is because we have our specialist, Dr. Cameron Jack, on with us today. Hello, Cameron.
Hello, thanks for the invitation. I'm more nervous than ever to answer specific OA questions.
Today, you are the chump,
Yeah.
Cameron, that everybody's trying to stump. So good luck with it, man.
That's exactly right. That's what I was going to say is, Cameron, we've actually brought you on today because you're going to be the chump, or people are going to try to stump you.
I feel like I should be more used to being a chump, but I'm still quite sweaty over here.
Well, Cameron, we get so many questions about OA, and Amy and I just kind of had these questions on our list for a long time. And we're like, Cameron knows about this stuff, so maybe we should just ask him to join us on 2 or 3 Q&As in a row. And we've got a ton of OA questions, so we're like, hey, let's just make it a 3-part series, and you be the guy who helps us out with that. So,
all right, well, I will do my best.
Okay, well, so this is the first of the 3-part series. The first question that we have today, Cameron, is that this person has heard of experimentation using oxalic acid with glycerin on a sponge. Is this to prolong a treatment period? Why would someone do that? And what are your thoughts on this?
Okay, so yes, you're right. The reason somebody would use oxalic acid, mix it with glycerin, so put it in something like Swedish sponge, is to prolong the period of treatment. Oxalic acid definitely can be effective at controlling Varroa. The thing about it is when it's applied according to the label, it breaks down very quickly. So it's like a good flash treatment. So people have been working to extend it to basically make it an extended release treatment that will last a bit longer. So now there are different products that are legal products that are being released out there, such as Varoxan, which is again kind of using glycerin as a carrier to kind of make a slow release kind of a treatment. People kind of before Varoxan had been also experimenting with this and using this in their colonies into a Swedish sponge. So now that there is a safe, effective treatment in the United States with Varoxan, and then other countries have also other products like Aloin caps. And so now that there are some other products out there, people really should be using what is, labeled in their countries and using these treatments according to the label. But yeah, the idea of it is to make it an extended release product. I have kind of mixed feelings about this. I am thrilled that there are products now that can be used to make oxalic acid effective over a longer period of time. My only hesitation maybe I would have with something like this is because right now we do not have any resistance to oxalic acid that has been reported around the world, and it's been used for a long time. And I think part of the reason for that is because OA has always just been such a flash treatment. It's there for, a few hours and then it's gone. But the way that we're making this, if we just get over reliant on these extended release OA treatments, then we definitely still have probably a greater likelihood of our varroa developing resistance to them. So we just need to be judicious and smart and not just over reliant on one method or one treatment for controlling varroa.
Cameron, I love that you said follow the label. We didn't even prompt you beforehand to say that, so it's nice that you said that out loud. All right, I've got the next
tattooed
question.
on my chest.
Good. Oh, well, I'll take your word for it.
We're learning so much about Cameron.
Yeah, we should have had you on earlier,
I
Cameron.
know we've known for you for how long, and learning about this just now.
And
I
OA
don't
's
show
on
it very much.
Yeah, that's on your chest and
Not
OA
my
's on your
proudest
back,
moment,
right? Yeah. All right. So next question: 4 grams of OA treatment. Now, again, only can follow the label, but how long does it last as an acid vapor in the hive? Their questioner said, is it 2 hours, 6 hours? Do you have any idea how long OA, when vaporized in the hive, lasts?
Yeah. Okay. So this is maybe me being a little bit of a chump. I have an idea here, but I don't know exactly, at the molecular level, like how much of this OA breaks down over time. I do know that we have done some trials where we put in like sticky boards to measure the mite fall like after we apply. And we've done those in like 24-hour increments. And we'll see the biggest drop within 24 hours, a little bit after 48, and then a little bit after 72, but basically nothing after 72. And so, applying like OA as a vapor, like you're describing here, the beauty of it is it coats kind of everything on the inside of that hive, but it just doesn't last for too long. So I don't really know kind of less than 24 hours how long it's lasting. My guess is probably it's not super long, just given what we have in terms of mite drops. It's probably in the first 12 hours or so is where it's doing the vast majority of dropping the mites. But I just haven't really gone into that level of detail yet to look for it. But we know that OA is very water soluble, especially where we're here in Florida with high humidity, especially in the summertime. I bet that it's starting to break down rather quickly, but we know that it works. The OA vapor is really going to work the most effectively in the first 72 hours. And then after that, the treatment is essentially gone.
All right. So for the last question of this series, Cameron, I've not heard of this one before, but some people will spray packages with an OA sugar solution. So they'll spray with plain sugar solution at first, so then they go through and I guess spray with OA. First of all, is this legal? And second of all, do you think this is an effective treatment?
Yeah, so good question. And it is a legal way to apply oxalic acid to packages. I mean, it is originally when the EPA released kind of the emergency use permit for oxalic acid, they mentioned this specifically, spraying OA sugar syrup to packages as a method of treatment. So it does work. It's not super ideal in my opinion, because you still have to spray a decent amount on these bees in a concentrated, area, and you'd still have to kind of bounce them around a little bit, I feel like, to get enough of a drop for the mites. You just got to make sure those bees inside the package are coated, and it's not super easy to get to all of them when they're all kind of bunched up in a package. So yes, it can work for sure. It's not my preferred method of using OA, and I don't think it does a really great job given the other options, even just trickling on full hive or vaporizing the hive, or using any of these extended release, or using the extended release treatments like Varoxan or aloe and caps. They're going to do a better job than just applying it to packages. I do know some beekeepers that will do this as they purchase packages and kind of bring them into their apiary. They just want to try to get them off on the right foot and make sure their packages are clean before they get them installed. But I feel like your bees, after like shipping and stuff and transportation, they're already quite stressed. It's just not usually something that I would recommend unless they really had levels of concern. If you're in an area, a part of the world or something where you're very isolated and you don't have, a lot of varroa in your area, then maybe that's something you would do. I just don't know that it's worth it, at least where we are, where we have high reinfestation rates with varroa. So you might as well get your bees set up and help them get established and then start worrying about varroa.
I think that's fair. Well, this was a very fun OA set of questions. Stay tuned for the next 2 that we're going to have as part of this series. And of course, listeners, when you have questions, if you have follow-up questions, maybe we can give Cameron even more series of OA. So you know how to find us, you know how to ask us questions. Don't forget to send us a question on either email or one of our social media pages.
Okay, well, thanks for having me on.
Hey everyone, thanks for listening today. We would like to give an extra special thank you to our podcast coordinator, Jeffrey Carmichael. Without his hard work, 2 Bees in a Podcast would not be possible.
Visit the UF-IFAS Honeybee Research and Extension Laboratory's website, ufhoneybee.com, for additional information and resources for today's episode. Email any questions that you want answered on air to honeybee@ifas.ufl.edu. You can also submit questions to us on X, Instagram, or Facebook at UF Honeybee Lab. Don't forget to follow us while you're visiting our social media sites. And thank you for listening to 2 Bees in a Podcast.